Hi, my name is Amr Jones, and I'm going to be doing my presentation on Chapter 4, the sensory cell. Sensory neurons are pseudo-unipolar with one extension from their cell body which branches into two directions. One end receives information while the other transmits information. Technically, sensory neurons lack structural dendrites, although the receiving end serves to be a functional one. Sensory neurons pack their cell bodies tightly together in a region called the dorsal root ganglion, which runs up and down the spinal cord. The dorsal root ganglion is near the spinal cord, and while sensory neurons don't send any information to their cell bodies, the information they're sending does pass by the dorsal root ganglion on the way to the spinal cord. The mesencephalic trigeminal primary sensory neuron is the only sensory cell that has its cell bodies actually located in the brain, and these cells receive information from the teeth about mastication, and it was a big source of conversation about evolution. Primary sensory cells receive many different kinds of information from the skin and muscles. Different kinds of sensory cells are receptive to different kinds of information, allowing for the diversity and differentiation of stimuli received. Some categories of sensory receptors include mechanoreceptors, which are receptive to pressure, proprioceptors, which are receptive to position, thermoreceptors, temperature, and nociceptors, pain or tissue damage. And the main difference in sensory cells is that the proteins which constitute their receptors are different based on gene expression. Receptors determine which kinds of stimuli cells will respond to, and these differences are due to differences in gene expression across these cells. And within the broad classifications of primary sensory cells, subtypes for the different receptive fields exist. So these are some examples of subtypes of mechanoreceptors throughout the different layers of the skin. And I found a study which investigates a possible interneuron in the spinal cord and a neuropeptide in a certain sensory neuron in mice, which possibly conveys positive touch and is responsible for encoding reward of positive touch in mice. And the receptor on the interneuron was called PROKR2, and the neuropeptide was called PROK2. And they used different methods, but they used transgenic mice, which were genetically altered, and they used biologic markers and viral tracers to investigate the gene expression, but also they knocked out PROKR2 and PROK2 in the ABL, which was the experimental condition. And then they tested the condition with these genes knocked out versus the normal control. They put them through a behavioral paradigm where that differed in pleasant touch. So one chamber received pleasant touch, the other didn't, and they compared them in that regard. And then also optogenetics were used to stimulate PROKR2 neurons in genetically altered mice compared to control. And then they measured the time difference spent in each chamber. And they also used behavioral and physiological responses measured in response to gentle touch across the gene ablation conditions. And they found that male and female mice in the control condition spent more time in the chamber where they received touch than the other chamber, and this was not found in the ABL mice. So with the genes removed, they didn't have a preference over if a chamber provided positive touch or not. And they also found the photostimulation of the receptor, the PROKR2 receptor, increased the time spent in that condition. And they found reduced heart rate, plasma corticosterone levels, and increased thermal pain threshold in response to gentle stroking in the control, but not in the ABL condition. And then there were also some consequences found for lacking the receptor and the neuropeptide releasing neuron. And this was less interest in novel stimuli and sociability. And that was my presentation. Thank you.